Bismillahirrahmanirrahim. Assalamualaikum everyone. Welcome to Stream of Knowledge. Today we will learn about plasmids. After hearing the term plasmids, the first question that will come in your mind will be, what are plasmids? As you can see in this picture, there are two terminologies that have been used, plasmids and bacterial DNA. Why the terminology bacterial DNA I am used with plasmids? Plasmids are actually extra chromosomal. What? What? Extra chromosomal? Bacterial DNA? Let us simplify it. As we know, chromosome contain DNA, and DNA contain the information about all the activities of bacteria. But plasmids do not. Plasmids have different genes. They do not contain those DNA or those genes that carry the activities of bacteria. That is why it's called extra chromosomal. And as we have discussed in our previous lectures about essential and non-essential components, and plasmids are the non-essential, meaning they are present in some bacteria, and bacteria can live without plasmids but cannot live without bacterial DNA. So, plasmids are the extra chromosomal, double-stranded, circular DNA molecules. As you can see here, it's circular, it's double-stranded, it's extra chromosomal, and they can replicate independently of bacterial chromosome. As I say, plasmids are extra chromosomal, mean they have different gene. But what kind of genes it carry? It carry gene for antibiotic resistance. Antibiotic resistance genes? But how? Actually, bacteria contain genes that form enzymes. And for example, beta-lactamase. We have already learned that beta-lactamase is an enzyme that breaks down the beta-lactam ring of antibiotics, for example, penicillin. So the bacteria, for example, Staph aureus, that contain plasmid will produce these enzymes. Another gene that it carry for are exotoxins. We already discussed that exotoxins are the polypeptides that are secreted by bacteria. For example, Antibiotoxins of E. coli. It also contains gene for Pili, which mediate the adherence of bacteria to epithelial cells. It also contains gene for the resistance to heavy metals and to ultraviolet light. It also contains gene for bacteriocins. What is bacteriocins? Actually, bacteriocins are the toxic proteins that are produced by certain bacteria. For example, E. coli produce bacteriocin called colicin. These proteins are beneficial for bacteria but harmful to the other bacteria. For example, bacteria compete for other bacteria for the food competition. If another bacteria try to colonize the E. coli site, then the E. coli will produce colicins, and these colicins will kill another bacteria, and it prevent the colonization of harmful bacteria. For example, E. coli is a normal flora of our gut, and it inhibits the other bacteria to colonize. Oh yes, our body have normal flora that is essential. Now, what are the types of plasmids? There are two types of plasmid, transmissible plasmids and non-transmissible. Transmissible plasmids are those plasmids that can transfer from one cell to another by conjugation. That we will discuss later. And non-transmissible that do not transmit from one bacteria to another. Yay, we have discussed all about plasmids. Thank you for watching. If you really like the video, enjoy the video, do subscribe, like, comment and share.